Sanger dideoxy sequencing is a sequencing method that is still used today, pretty much in the same fashion from when it was first designed, with some updates. This lecture will discuss the original as well as the updates. We've previously talked about oligonucleotides and how some specific nucleotides are incorporated into oligos. One of the things that Sanger dideoxy sequencing can do, it can label DNA with a radioactive marker. Now, there's two different ways of labeling DNA with a radioactive marker. One of the ways is having a P32 label, a radioactive phosphorus right here, that an enzyme called a kinase will remove from something like a DATP and put that most outlying phosphorus, the gamma phosphorus, this is alpha, beta, and gamma phosphorus, transfer a radioactively labeled phosphorus to a short stretch of DNA to radioactively label that. Another way of doing it is to incorporate an S35 labeled nucleotide, in this case S35ATP, and that gets incorporated into a growing chain of DNA. In any event, you need to, in the original Sanger dideoxy sequencing, you need to radioactively mark your DNA somehow so that when you put an X-ray film against the gel, that you're able to see the different patterns of your DNA. Another nucleotide that is used is a dideoxynucleotide. So remember, a regular nucleotide is a deoxynucleotide, but a dideoxynucleotide is missing both oxygens, not just one, so it is unable to have another base added to the 3' end of it. In other words, there's no oxygen for an incoming phosphorus to add on to. So when a dideoxynucleotide is incorporated into a growing DNA chain, nothing more may be added to that chain. So here's generally how it works. You have to have a known DNA sequence that is next to your unknown DNA sequence that you want to know the sequence of. And you make a primer. You make a short oligonucleotide stretch that will bind to your known DNA sequence. So remember, if your template strand is 5' to 3' in this direction, then your primer is going to bind 5' to 3' in the opposite anti-parallel direction. And you will anneal that primer to that known sequence on your DNA right next to the unknown DNA sequence that you want to know the sequence of. Then you will divide up this annealed stretch of DNA into 4 different reactions. In one reaction, you will have S35 labeled DATP, DTTP, DCTP, and you will have dideoxy GTP, as well as the polymerase that's going to add your base and a certain amount of salt and a good buffer and all the normal things you would have in a reaction. But the important thing is, in a G reaction, you would have dideoxy G added. This means that every time there is a C in the template strand, you're going to have a certain possibility of adding a dideoxy G. But remember, there's also some regular GTP in there as well. So what that means is that maybe 1% of the time you add a dideoxy G in this position, and that's going to not allow, if it's a dideoxy, it's not going to allow anything else to be added on after that. But 99% of the time, it's a deoxy G, and then you can continue on to the next G. So again, 1% of the time, a dideoxy G is added. 99% of the time, it continues on to the next G. These A's represent the S35 labeled DATP. In other words, now we have a radioactive strand right here that we can see on a gel. You have to have an S35 DATP add. If you don't have radioactively labeled ATP add before your first dideoxy, you're not going to be able to see that first G at all. So, getting back to all the strands that were made. Every time there is a C in the reaction, there's a certain probability of adding a dideoxy G, and then most of the time you're adding a regular G and the reaction continues. But even if 1% of the time the reaction stops, you're going to have a large number of differently sized pieces of DNA, each size representing the ending with a G. Then you can run those on a gel, and you're running them on a denaturing gel so that they're running only according to size. So if you're running a G lane on here, you'd see, something that's 26 bases, and then something that's 27 bases, and something that's 32 bases, etc., and 41 and 49, etc., all the way up. So basically you'll have just a bunch of fragments here, and all the sized fragments will correspond to where there was a G ending in the sequence. Then, in your A reaction, you're going to have not only your S35 DATP, but your dideoxy ATP. Remember, the very first A, you have to have an S35 DATP to be able to see that strand at all. But then after that first S35 DATP is incorporated, every single A after that, you're going to have a certain possibility of adding a dideoxy ATP. With 1% of the time, a dideoxy ATP adds. 99% of the time, it continues. So basically, you get a bunch of strands, each ending in an A, representing every place in the sequence that in the template sequence there was a T, so that every time in the template sequence there was a T, you're going to be adding an A in your growing chain. Then if you, on the gel, run the A's next to the G's, they're going to be characteristically sized fragments every place there is an A. Then you're going to do the same thing for the T and the C reaction. The T reaction has the dideoxy T's. The C reaction has the dideoxy C's. And when you run all of those on the gel, you're going to see the same thing. So every place there was the sequence ending in G, you're going to get a mark in the G lane. Every place there was an A, you're going to get a mark in the A lane. Every place there's a T, and every place there's a C. And then you're going to read the gel, and it's read from a 5' to 3' direction from the bottom. So here you'd say this is C, T, A, C, G, G, A, T, T, C, G, etc. from the 5' to 3' direction, reading all 4 of your lanes, whichever one is next in turn. This is a graphic which sort of shows the whole step. This one shows it using a radioactively labeled primer instead of the S35 DATP, but the result is the same. You have the fragments, each one ending with a particular sequence. You've run them all on the gel. Different order of loading rather than G's, A's, T's, and C's as I showed you. This gel was loaded as A's, C's, T's, and G's, but you're still reading from the 5' to 3' direction, reading that new sequence that is being created, which happens to be the opposite of that original template strand. So you just need to make sure that you understand which sequence you're talking about, that when you are talking about the newly created strand that you're reading the sequence of here, that it's the opposite sequence of the template strand that you were just sequencing. Now, some technological improvements that have happened. In gels that were using radioactive labels, you actually needed to be able to load 4 lanes, one for the G, one for the A, one for the T, and one for the C, because basically the radioactivity just simply made a black mark on the X-ray film. So you could not put them all 4 in the same lane because you would just have a series of these little black marks going all the way up. So the technological improvement that happened was hooking a fluorescent tag to each of these dideoxynucleotides that are added. So dideoxy-G is one color, dideoxy-A is a different color, dideoxy-C and dideoxy-T. They each have their own separate color. This means that you can now do capillary electrophoresis. So rather than having to run them on a gel and put an X-ray film against the gel, you can now just simply load them into capillary electrophoresis. They separate out by size. As each of those sizes passes in front of a small window of the detector, and remember, they're still all one base separate in terms of their size. So as they pass in front of that little window, each fluorescent dideoxynucleotide is detected by a laser, and this makes something called a chromatogram for you. And so that chromatogram then gives you the sequence of A, C, T, etc. So basically, if you were the laser and you're reading it, you'd come along here, and as each of these things are moving in front of the laser, you'd say, ooh, a little bit of green, ooh, a lot of green, and now less green, and ooh, a little bit of blue, oh, more blue, and then less blue. And so that's what's making these peaks right here, as each one of these passes in front of the laser. So this is what it would look like as it's passing, size by size, for example, if you had this particular sequence. And then here's the corresponding chromatogram. And so rather than taking up 4 lanes in a gel, you can now have each one run in turn on a capillary electrophoresis, and you can be much, much more efficient. So you can be running multiple samples at the same time, and you can actually get much longer reads. As you go up the gel, these bases actually tend to get a little closer to each other in terms of size. Their sizes are a little bit close to each other. And so you have resolution problems as you start to get past maybe 150 to 200 bases if you were using one of these older radioactive gels. But the capillary electrophoresis is actually able to separate it out much better because it's not read until the size actually comes down here. So it's actually able to separate them better. So you can easily get reads of 600 to 800 bases off of each off of each sequencing reaction. So not only are you more efficient by being able to use capillary electrophoresis, and instead of having to use 4 lanes, only use 1 lane or 1 capillary at a time, you also get a much longer read out of them. Now, this does not include next-gen sequencing. That is a separate lecture. Here's an animation of the process if you're interested.